Okay, ladies and gentlemen, so you should have in your possession these genetics notes. This should be such a review for you. You should be able to whip through them no time flat, and eventually by Friday, Monday, we're going to start working in the big packet that you've gotten. So we're going to whip through these notes really quickly. If you have any questions, let us know. I'm hoping that, and Miss Lum and I are both hoping that you just know this stuff. So real quick, let's just hit it. Genetics, you know, is the study of how traits are passed from parents to offspring. Offspring is another name for the babies. Heredity is that the traits are passed from one generation to the next. You know sex cells already because we did the meiosis bead lab. So we are the making of sperm or egg, which are gametes, which are also known as germ cells. The sperm will have half of the genetic information. It is a haploid number of N. The egg would have half the genetic information as a haploid number of N. This gives you one individual, which is a diploid 2N number. Once egg and sperm join, we know we have fertilization and it is a zygote and the chromosomes are going to pair up. In humans, in their body cells, also known as somatic cells, there are 46 chromosomes, which is the diploid number. They got 23 from mom and 23 from dad. If you are a fruit fly, the sperm had 8 haploid number, the egg had, the sperm had 4, a haploid number, the egg had 4, a haploid number, giving us a diploid number of 8. That would give you a fruit fly. Every chromosome has a lot of locations on it called genes or loci or locus, and they carry the genetic information that's different. This would be the segments of DNA that we looked at, I don't know when, for protein synthesis. 6 terms you have to know. These are words that should be as common to you as hello and goodbye. Dominant, the stronger of 2 genes in a gene pair. The trait will be expressed or represented by a symbol. In this case, a symbol like capital B would equal black. Recessive would be the weaker of 2 genes in a gene pair. It will only be expressed if there are 2 recessive traits. Little b, little b, little b would be white. That's the only way that you would get a recessive trait showing. Homo, prefix, means the same. If you have 2 letters, one from mom and one from dad, that are the same, the trait is the same. Big B, or little b, little b. Big B came from dad, big B came from mom, little b from dad, little b from mom. These are pure traits. They are also known as homozygous. Hetero is different. This is also known as hybrid. Both of the traits are different. So mom or dad gave a big letter, mom or dad gave a little letter. Don't forget, these are just symbols representing traits, representing genes. The symbols themselves, the letters that we're using to represent the traits, are known as the genotype. Big B, big B, big B, little b, little b, little b, they are the genotypes. Phenotypes are a description in word, like a photograph, PH, photo, pheno. These are the description in words. Is it black? Is it white? Is it brown, blue, tall, short, freckles, no freckles? So those terms you absolutely need to know for when you are starting to do genetics problems. Other terms that you should be familiar with. P1 stands for the parental generation. The first parents would be P1. P2 would be the second parents. F generations, the first filial generation. Filial means basically the babies of the parents. When 2 F1s mate, you get P2s, and then they would make F2s. F2 is the second filial generation, the babies of the first generation, and that would continue on and on and on and on. You could have F3s, F4s, F5s. Something that you should be familiar with is Punnett squares. This is the method for determining possible offspring with statistical accuracy. You should remember Mendel, the father of genetics. He was the Austrian monk. He was working with pea plants. You're going to need to go to your coloring to come up with some of the traits that pea plants and Mendel worked with, so you will know what is dominant and what is recessive. The basic Punnett square is that box of 4. You have your traits of the parents distributed on the top and the bottom. You're separating them one letter, one letter, one letter, one letter, and then you put them together to get the possible offspring in each of the boxes. The possible offspring give you 25%. The whole thing would be 100%. These are now your F1 babies. Flip side of this is how to do a genetics problem. So in order to do this, we have 5 steps that you always need to show. When you get to the big packets, you're going to see that the first thing we're asking you to do is write a key. You're going to determine the letter that indicates what trait you're talking about, and then you are going to actually always write big, 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 little, little, little. If you use them or not, write all three options. Big T in this case would be a homozygous dominant tongue roller. Big T, heterozygous tongue roller showing the dominant trait. Non-roller would be little t, homozygous pure for the recessive trait. The next thing you're going to do is write the cross. Who are the parents? So in this case, it tells us that big T times big T are the parents that we are going to use for our cross. These are the P1s, the genotypes of the parents. Step 3, draw your Punnett square. Parent 1, big T gets separated on one side of the box. Parent 2, big T gets separated on the other side of the box. Then you're going to actually complete your Punnett square, put it together, come up with your genotype ratio, sometimes referred to as the GR. Genotypes are the letters that are possible. You go back to your key, big T, big T, little t, write them out horizontally. How many of the babies are big T? 1. Big, little, 2. Little, 1. You need to show both the letters and the numbers of how many in a ratio format. The phenotype ratio is going to be the 5th step. Here you are writing out the possible words. Again, go back to your key. Rollers versus non-rollers. How many of the offspring are rollers? 1, 2, 3. It doesn't matter if they're homozygous or heterozygous, they are rollers. We have only one non-roller. Again, you have to write out the words and you have to write out the ratio in numbers. Okay, so hopefully that was the quickest review of genetics, and all of these things sound really familiar to you. If not, let us know and we'll try to give you some more updates on how to help you through these problems. Tomorrow we're going to go through some of the special types of problems, and then you'll be able to get into your packet. Have a good day.